For those who joined us yesterday, thanks for coming back again. And for those who are joining us for the first time, welcome. And this is a careers and science mini conference for people in secondary school or high school thinking about what they might do in university or after they leave school. So as you're all joining, I'll just recap what we had yesterday. So we had many different talks. We both had people who had been working in science for quite a long time and were established group leaders. We also had more junior scientists doing their PhDs or research assistants or research coordinators. And you also got to have a look inside some of the labs at the UK DRI in some of the Follow Me Around the Lab videos. A lot of people asked yesterday about whether the recordings of these sessions will be available, and yes, they will be, and they'll be here on this YouTube channel from the UK DRI. And you'll also see there that we have recordings from the same events that we ran in 2021 and 2022 with a completely different lineup of speakers. And so even if you've enjoyed these sessions and you want to find out more, you'll get a lot of different insights and different advice from those videos if you want to take a look back at them. And in a couple of weeks, the recordings from yesterday and today will be there as well. So a reminder for those who were with us yesterday and some insight for those who are just joining us for the first time. We are coming to you from the UK Dementia Research Institute, and you can see on the map on the right side that this is an institute spread over many different centres within the UK, including Edinburgh, Cardiff, Imperial, King's and UCL in London and Cambridge. And we're all focused on dementia. And what is dementia? Well, dementia is an umbrella term for the loss of memory and other thinking abilities severe enough to interfere with daily life. So we think of dementia as a symptom of a disease rather than a disease itself. And some of the diseases that cause dementia are Alzheimer's disease, vascular dementia, frontotemporal dementia, and other diseases. And scientists research these complex disorders using a variety of techniques, some of which are kind of depicted here in these little cartoon images. And we use modeling like animal models, cell models, or postmortem brain tissue. And we also use more clinical research like MRI scanning or looking in people's blood. So you're going to hear a lot more about our research and our careers today. And this is the lineup for today's session. So we're going to start with a talk from Professor Tara Spires-Jones in just a minute, but there will also be some more flash talks and Follow Me Around the Lab videos to give you quite a varied insight into how the Institute runs and what the science we're doing is. So just to kind of catch up with you guys that are writing into the chat, we've got, as you can all see, quite a huge number of places being mentioned, and it's so great to have such an international audience. But this does mean that kind of the systems for school and education are going to differ quite a lot across these countries. And so we're going to give as much insight as we can, but as you'll know, lots of our speakers come from specific countries and are only familiar with one education system. So our advice might be a bit more general than maybe perhaps what you might need to do for your exact A-levels if you're in the UK. So I think that's everything that I need to cover to start us off. So I want to introduce our first speaker, which is Professor Tara Spires-Jones, coming from the UK DRI in Edinburgh. And Tara is a principal investigator studying synapses or neuronal connections in different neurodegenerative diseases. So over to you, Tara.
Thank you so much. And I hope everyone can hear me and see the slides. So as Cliona mentioned, I'm Tara Spires-Jones from the University of Edinburgh and the UK DRI there. And what I'll talk about today is in three parts. I'll talk a little bit about the past, so introducing you to neuroscience. Cliona already introduced you very well to what dementia is. I'll introduce you to Alzheimer's disease specifically and what's known about that a little bit in the field already. Then I'll talk a little bit about what we're doing right now in our research group in Edinburgh. And here we are looking very shiny in front of McEwan Hall, where you graduate if you happen to come to Edinburgh to work with us. And then I'll spend the last couple of minutes talking about what I think is the future of dementia research and the pathway towards effective treatments for people living with diseases that cause dementia. So first, Why am I a neuroscientist? Why study the brain? So this is taking a step back from dementia and just talking about the brain. And why do I study the brain? It's because it's amazing. Your brain is absolutely phenomenal. So you'll all be aware of where it is in your heads, what it looks like roughly. This is a brain that's been dissected after someone has died and taken out of the head. But when you really start digging into it, it's phenomenal. So we have about 100 billion neurons, which are the brain cells that form the network in our brains. And those are connected by 100 trillion synaptic connections. So if we look at the brain in a little bit more detail, this is now a brain section from someone in a coronal plane like this. So you can see the gray matter along the outside, and that's where these neuron cell bodies mostly live. And then you see the white matter. And the white matter, one of my favorite fun facts about why the white matter is white, is that the white matter is where the neurons send their beautiful long axons to talk to other neurons. And because they're electrically conducting signals, these axons are actually wrapped in layers of fat called myelin, and the fat is white, and that's why your white matter is white. So you have the neurons living in the gray matter. They send axons through the white matter to talk to other neurons in other parts of the brain. And where they talk to each other is this synaptic connection, the thing Cliona was telling you that we study in our lab, called the synapse. And this is the presynaptic terminal where the end of one neuron is sending a signal, and the signal, the neurotransmitter, is actually inside these little bitty neurotransmitter, these little synaptic vesicles that you can see here. They're full of neurotransmitters. And when the electrical signal comes down the axon, these fuse with the presynaptic membrane and go and attach themselves to receptors on the postsynaptic side here. So this is the postsynaptic side of the synapse that's receiving the signal into the next neuron down the chain. And I want you to just think about the scale here. This is a centimeter in this scale bar, and you know how big approximately your head is. When you get to this level, we're now looking at 0.001 millimeter scale. So synapses are absolutely tiny. And you have 100 trillion synapses in your brain. That's more synapses in your brain than there are stars in our galaxy, which is my other favorite fun fact about the brain. So you know all about what dementia is now, this umbrella term, so I won't tell you about that again. I'll start to focus in on why we focus in on dementia. Why do we study it? I can't see a show of hands here because we're on a webinar, but usually if I ask even a group of people in a younger age group like yourselves, if you've met someone with dementia, there's a pretty substantial proportion of the room that may have met someone living with dementia. And that's because it's so prevalent. So there are about 9.5 million people in the Americas, 12.5 million people in Europe, and overall worldwide, over 50 million people are living with dementia. So it's a big issue because these are very devastating neurodegenerative diseases that cause dementias. And why do so many people have dementias? It's not exactly clear. So like you already know now, dementia is not a single thing, it's the symptoms, and there are lots of different diseases that can cause dementias. But what we do know is that if we look at all of the dementias, there are three big things that sort of contribute to whether or not you will develop dementia. Age is the biggest risk factor for most dementias. Genes, so things you inherit from your parents, can increase or even decrease your risk for dementias in some ways. And then there are lifestyle factors that we can all control ourselves that can modify our risk for dementias. So aging is something that hopefully will happen to all of us. My mother always says it's better than the alternative. Genes are something that we are born with, and we can't at the minute really change those, but we can control our lifestyle factors. So when I'm talking to young people or older people or anyone actually, I like to tell you things that we can all do to change our own risk for dementias. So it's estimated that about a third of people who end up with dementia could have perhaps prevented that by changing modifiable risk factors, these lifestyle factors. It's really not to blame anyone who ends up with dementia because you can't control the other 2/3 of the risks at all. What I'm trying to tell you is that it's a good idea to take good care of your brain. So you can reduce things that are associated with increased dementia risk. So things that are bad in terms of dementia risk are smoking, high blood pressure, physical inactivity, and hearing loss and social isolation. And things that are associated with reduced risk are things like exercise, healthy diet, staying in education, so well done you for a late afternoon listening to a lecture, and keeping both physically and socially active. So we don't exactly know how these risk factors lead to dementia, and some of them might actually not be direct links, but we do know that it's probably worth your time to take good care of your brain, avoiding head injury, keeping yourself fit, and that will hopefully reduce your risk of dementias. What's happening in the brains of people who have dementias? So our lab focuses on Alzheimer's in some of our projects, so I'm going to talk mostly about Alzheimer's today. And Alzheimer's disease is the most common cause of dementia and one that I would imagine most of you have heard of. It was first described by this chap here, Alois Alzheimer, way back over a century ago, and he was describing the symptoms of his patient August Dieter. And she had the symptoms that we all are familiar with today of severe memory loss. She didn't know people, she didn't know herself, she said, and it got worse and worse and worse as her life ended. And what Alzheimer discovered when he looked at samples from her brain after she died is that three cardinal features that we still use today to define Alzheimer's disease. And these are what we call atrophy or brain shrinkage. You can see that here. This is someone who died of a non-neurological or non-brain disease. Compared to the size of the brain of someone who died with Alzheimer's disease, you can hopefully appreciate the shrinkage. So that's because the brain is literally dying. Brain cells are dying over the course of this disease. And we call that atrophy. And Alzheimer was able to take advantage of, at the time, a really fancy new technique called microscopy. He didn't have yellow and green, but he had silver-stained sections from August Diezer's brain. And he described the accumulation of two big pathological lesions in the brain called A-beta or amyloid plaques. These are clumps of amyloid beta peptide outside of the cell bodies that is just in the neuropillar, what we call it, outside of the cells in the brain. And he described these clumps of tau inside, or clumps of neurofibrillary tangles inside neurons in the brain that we later learned were made of tau protein. But these two pathologies, plaques and tangles, don't show up all at once in the brain. We know that this is a very slowly progressing disease. And for those of you who have met someone with dementias, you'll know that it takes years for their memory to decline and their behaviors to change and their thinking to change. What we know now is that in the period even before you have any symptoms whatsoever, people who are on the path to developing Alzheimer's disease are accumulating these amyloid plaques, these are that are in red and yellow here. And at the time when you start to have detectable symptoms, when a memory test could see that there's something a bit wrong, you start to really accumulate more of these tau tangles and they start to spread through the brain. And that's also when you start to really lose a bunch of your brain cells, your neurons, and all the way through the course of the disease, you're losing these synapses. And it's, in fact, the loss of these synaptic connections is the strongest correlate or the strongest tracking with the cognitive decline that's seen in the disease. One other bit of scientific background I'll give you is that those tau tangles don't just appear in the brain and all over the brain, they appear and they spread through the brain. So this is a seminal paper by a guy called Heiko Brack, and we still use his staging scale to stage Alzheimer's disease today. And what they discovered is that if you look in very early stages of Alzheimer's disease, you have barely any tau pathology, only a tiny bit, and a part of the medial temporal lobe important for memory. But as the disease progresses, this tau pathology starts to spread through the brain, and eventually at the very end of the disease, you have tau pathology almost all over the brain. And what's interesting about this is these regions are connected by synapses that the tau pathology spreads through. So that's a bit of the background. So we know Alzheimer's disease is the most common cause of dementia. There are a few risk factors, and we know the pathologies that accumulate in the brain. What are we doing right now in terms of Alzheimer's disease research is we're looking at why synapses die and can we hopefully prevent that. And I'll give you just a little flavor of what we do in the lab. I know you have actual videos of like official ones, but if I can get rid of my laser pointer, I might be able to play this. So I've made a little video of our lab. Oh, I'm going to turn off that. So I made a video in our lab of one of the ways we look at synapses. So what I'm doing here is taking a small piece of brain tissue that was donated either by someone who died with Alzheimer's disease in this case, or we can take from the animal models that Cliona mentioned. And we take these little bits of brain tissue and we make them into even smaller bits where we can see all 6 layers of the neocortex. So all the way from that white matter to the top of the cortex. And I'm making little bitty chunks here. And then these are fixed in a special fixative, formaldehyde fixative with a little bit of sugar in it for just a few hours. And the reason we make the sample so small is so that fix can get in. And then here we're embedding these samples in a plastic resin that's liquid at room temperature when we start, but after we bake it, and that's a little label to show you which part of the brain it was and who it was from, after we bake it, we have this really hard plastic resin. And the reason we put tiny samples of brain and really hard plastic is so that we can use this machine and this diamond knife that's in blue here with a little diamond, there's a shiny bit on the end, to cut ultra thin sections. And this is because synapses, as you saw from that scale bar a few slides ago, are absolutely tiny. And they're so tiny that we can't look at them accurately with standard microscopy techniques. So we need to do this. So now here we are sectioning bits of the brain into ultra thin sections, and you can see there's serial sections, which means it's like you're slicing off a block of cheese. So you're getting adjacent sections, and we're able to build up a 3-dimensional reconstruction of 70 nanometers, so very thin, smaller than a single synapse section. And those little thin sections are now on this cover slip that we're staining with an antibody. So the antibodies are very powerful. They can bind to very specific proteins. So for example, here we're binding to that amyloid plaque protein, as well as proteins that label the synapses. And here's Dr. Rosie Jackson, who made the video, and she's now showing you how we take images then of these now glowing proteins on the slide so that we can look at individual synaptic connections. I'll let the video run for a second here. I'm just looking at my time. But it is absolutely one of my favorite things to do in the lab is cut little brain sections and then go in and image them on our microscopes. They're absolutely beautiful. So Rosie's now taking a picture here of all those individual sections. You can see them appearing here. And then she zooms in with a very high resolution objective and finds an individual plaque. There's the plaque there. And then she'll go along the section and take a picture of that plaque and all its surrounding synapses. And with this technology, we're able to ask the question of, is that plaque peptide, amyloid beta, inside single synaptic connections in the brain where we think it might be toxic? So the first question we asked with this technology, many people before us had shown synapse loss in Alzheimer's disease, but we were able to replicate this. Replication is a very important part of the scientific process. So with this new technology, we were able to stain presynaptic terminals in magenta here and postsynaptic terminals in cyan here. And we get these little opposed synaptic pairs that you can see here that we call snowmen in the lab because they look a little bit like snowmen on the microscope. And we were able to confirm that with this technology, we can detect the synapse loss in Alzheimer's disease compared to control patients. And we were further able to look in more resolution. So the novel bit was being able to look at where the synapse is lost and are they inside, are they containing synaptic proteins. So here we're showing you an area of the brain that didn't have either of the pathologies, an area of the brain that had one of these big neurofibrillary tau tangles, and an area of the brain with an amyloid plaque. And a really striking finding is that the synapses, I don't know if you can notice here, but the magenta and the cyan in this picture are completely gone inside the plaque. So we have this really substantial synapse loss right around the plaque pathology. And we can then use this high resolution to look inside individual synapses. And we see that both amyloid beta and pathological types of tau clump up inside individual synapses. So here the amyloid is in white and the tau is in yellow, and the synapses are still these pairs in magenta and cyan. And you can see it's not all the synapses, but in some of them we do have these pathological proteins. Further, we're able to look at these pairs and look at whether things like tau, I told you that the tau pathology moves through the brain, and we were testing the hypothesis that the tau is moving through the brain by actually jumping from the presynaptic side to the postsynaptic side of that communication, that synapse. And we were able to show that with this kind of technology. This is an example of tau inside the presynapse only, tau inside both sides of the synapse, and tau in the postsynapse only. We were able to show that in human brain and in model systems, we do see tau accumulating in these pairs, and that in the animal models, we actually can see that it jumps from pre to post. So I think I'm still on schedule. So I'll finish up with future. So how are we going to actually move from this really interesting and exciting fundamental neuroscience that we're doing on microscopes on tiny bits of tissue in the lab, which I find amazing and fascinating because I'm a neuroscientist, but how are we going to take that and make it into something that can help the people who are living with Alzheimer's disease and other dementias today? So one way we do this is the discoveries that we make in the lab. We then work with colleagues in industry. So here's one example of that. This is a person named Susan Catalano who came to me at one of our big scientific meetings. One of the fun things about being a scientist, if you're considering it for a career, so we get to go to big meetings all over the world and present our data on posters. So I had a poster at the Society for Neuroscience meeting one year in, I think, 2012. So you were probably very young. And Susan came to my poster and asked if we could talk about this idea of oligomeric A beta, or that amyloid plaque protein and peptide accumulating in synapses around plaques. And we worked with them for a few years. We looked at whether a drug that they had developed that knocks amyloid beta off of a receptor at synapses, they were testing it in mice. We had a look at it on our human tissue. And now this company has this drug that we help them in a very small way to move forward, is now in clinical trials being tested to see if it will help people with dementias. We don't know yet. The trial's not finished. And a disclaimer, now I'm on their scientific advisory board. So we work with industry to try and take the discoveries we make in the lab and try and move those forward so that we can eventually help people living with dementias. And I have to say, none of this is my work, but as a field, it is an exciting time to be an Alzheimer's disease researcher. For many years, we had many clinical trials for Alzheimer's disease that all failed for about 3 decades, if I remember correctly. But now there have been a couple of new drugs approved. They're not perfect, but they have been proven in clinical trials to slow disease progression. So they slow the cognitive decline or the decline in your thinking abilities a little bit, but still for us as scientists, it's satisfying to know that something that we've worked on, like this amyloid plaque peptide and how we think it's toxic to synapses, these new drugs are actually removing that amyloid from the brain and they're slowing slightly the disease progression. So I want to end with the idea that we need your brain to help solve some of these big neuroscience problems. And I've just shown here a little, sorry, Zoom's in my way here. I'm just showing you what one example of a scientific career can look like, just as an example if you're thinking about coming into neuroscience of one path that you could take. So this does show you my age in a way because it shows you when I was finishing high school, which was 1994. And I was doing that in Leander, Texas. And then I went to the University of Texas at Austin. I've got a picture here as an undergraduate, but I spent a year at Université Paul Sabatier in France, which was amazing. Graduated from undergraduate school in 1999 and got a scholarship to go to Oxford to do my DPhil or PhD with Professor Sir Colin Blakemore, looking at Huntington's disease models and the development of synapses. And that's where I really fell in love with the synaptic connections in the brain. Finished that training, so keeping in mind this is all still university training, finished that training in 2003 and started in January of 2004 as a postdoc, which is sort of even more training, but you get paid a bit better. So postdoctoral training in Boston at Harvard Medical School, MGH, and there's a picture of the building I worked in for 9 years here. And I was there for 9 years as a postdoc and then a junior faculty member, and then was going up the ranks a little bit and then decided to come back to the UK, came to Edinburgh in 2013 as a reader and was very excited to be promoted to professor in 2017. And then this is again a picture of us here in Edinburgh in the lab. So, oh, my Zoom's not working. Oh, just to also note that you can be a scientist and have a life. That in that time in Boston, I had my 2 kids and really have enjoyed the career that allows a little bit of flexibility to spend a lot of time with them. So I think I'm just about on time, and this is almost my last slide. This is my take-home messages for you. Your brain is absolutely amazing. Please keep it healthy. Try your best to not have head injuries, good lifestyle choices, and that will not only be good for your whole body, but it will also hopefully slightly reduce your risk for dementias when you're older. Our group has used lots of different approaches, but I showed you one called array tomography to make progress in understanding how the amyloid and the tau pathologies clump inside synapses and damage them in Alzheimer's disease. And for the future breakthroughs, we really do want the best minds. So again, do please think about becoming a neuroscientist. I love it as a career. I think it's absolutely fascinating. We get to see things that no one's ever seen before down microscopes, talk to lovely people like you, travel around. I'm right now in Manchester, coming to a different scientific meeting. So please do think about it. And I'd like to thank all the people in our group. This is my colleague Claire Durrant, who has a group in our department, and all of our students and postdocs and technicians who work with us. And we have lots of people that we work with around the world. So just to end by saying thank you to all of them. And I look forward to the Q&A, which hopefully I'll be able to attend, even though I have to switch venues in between.
Thank you so much, Tara, for such a great talk. It's really nice to see that kind of looking at the really tiny synapses and even what's inside them all the way up now to the progression of these drugs for Alzheimer's disease. So thank you so much for bringing us through that. If anyone has questions for Tara, please put them in the Q&A box, and we're going to address all the questions at the end of today's session. So throughout the session, any questions for any of our speakers or kind of any more general questions, please do put them in the Q&A box. So next we have an insight into one of our labs. So we're going to play one of the Follow Me Around the Lab videos. And this was made by Lais and Christina, who are based at the University College London UK DRI Centre.
Welcome to the UK Dementia Research Institute. This is the home lab, and we study the immune cells of the brain.
So thank you so much to the Hong Lab at UCL for making that video. I think it's a really nice example of kind of the ups and downs of everyday life in the lab, including when you accidentally drop your samples, which I thought was quite funny. So thanks to those guys. So next we're going to have 2 flash talks. These will be by Elena Veleva and Dr. Peter Swann. So first we're going to have the talk by Elena. So Elena is the Biomarker Factory Coordinator based in University College London at the UK DRI. So I'll let you have the floor, Elena.
Hello, let me just share my screen. Okay, thank you. So hello to everyone. My name is Elena, and as Cliona said, I'm the facility coordinator of the Biomarker Factory. Today I'm going to talk to you about a little bit about my career journey and what the Biomarker Factory actually is and what is my role in it. So let's begin with my career journey. I have graduated high school in Bulgaria in 2011, and I moved to the UK in 2012 to study biomedical sciences as a bachelor's degree, and I was working part-time in retail while I was doing that. When I graduated from my degree, I started working as an assistant technical officer at King's College Hospital. And my role there was to receive specimens from the hospital, book them on the system, and direct them to the correct clinical lab in the hospital. A year later, I was promoted in the same place. And then my role involved more of the pre-analytical processing of samples, so preparing samples for the actual analysis. In 2017, I have joined the Fluid Biomarker Lab at UCL as a research technician, and my role was to receive and process blood and cerebrospinal fluid samples for many different studies, store them in freezers, and database all the information about those samples. And in 2021, I have started my current role as a facility coordinator at the Biomarker Factory. So what is the Biomarker Factory? This is a facility that is funded by UK DRI, and it is part of the Fluid Biomarker Lab. And what we do is we measure biomarkers in blood and cerebrospinal fluid to detect normal or abnormal biological processes. And what is the purpose of the Biomarker Factory? It is to measure biomarkers in fluids in large studies. And we have the specialized equipment to do so. And our lab is led by Professor Henrik Zetterberg and Dr. Amanda Hesselgrave, who are experts in the field. So they work with many collaborators, and they provide expert advice that will hopefully help for the development of blood tests for diagnosing Alzheimer's disease and other neurodegenerative diseases. So what is my role in the lab? My role is more admin-based. So I'm involved in discussions with researchers. I speak to staff at all levels, both internally within UCL, different departments within UCL, other universities, and also commercial companies. And this means institutions both in the UK and internationally. I'm involved in the organization of projects, from planning to organizing shipments and placing orders for the reagents that are required to test those projects. I do the admin work, that is the generation of quotes, invoices, and payments for those projects. And I deal with the stock management of lab supplies. So I make sure we have sufficient supplies to test a certain project. And also sometimes I actually run experiments as well. I'm also involved in the implementation of new software. So recently we have I've joined a group that we have started developing a website that will help researchers request test measurements online. So this will make the whole process more streamlined. And what is a typical day in my role? I'd say it's different each day, but it mainly includes meetings, answering emails, record keeping, and general organization of the lab. And what I think are the most essential skills to develop in your career in academia are to have excellent organizational skills and time management skills, and also to have good communication skills, so to build a good professional network of people that you know, and to be a team player. And of course, nothing is possible without a good team. So I want to thank my team for their hard work, and especially my line manager, Dr. Amanda Hasselgrave, for her support through the years. Thank you very much for listening. If you have any questions, please contact me at my email address. Thank you.
Thank you so much, Elena. And Elena is not able to join us for the Q&A today, so if you do have any questions, please do contact her by email. And maybe, Elena, could you just put maybe your email in the chat again, just so people have access to that. Thank you so much for a great talk. It's really nice to see kind of a different side of science and a more supporting role, and potentially kind of, if people are not necessarily interested in medicine or necessarily interested in lab-based research, there's all these different types of roles around supporting science that are really important as well. So next, we're going to have Dr. Peter Swann give a talk. He's a clinician scientist based at the University of Cambridge. So over to you, Peter.
Hello, can you hear me okay?
Yeah, it looks good as well.
So my name is Peter Swann, and I'm going to talk about being both a clinician, so a medical doctor working in the NHS in the UK, whilst being a researcher in dementia. So I'm going to tell you a bit about what I work on, and then what my sort of typical week might look like, and then tell you about the sort of career journey I took to get here. So first of all, what do I work on? So it's dementia research, but particularly looking at inflammation. And there's more and more evidence now that inflammation, so the actions of the immune system, play a key role in the development and the progression of dementia and neurodegenerative disorders. And this can be inflammation in the brain, so neuroinflammation, with these specialized little cells called microglia that respond to injury or damage and become these little amoebas that go to the site where they're needed. And they can actually end up potentially doing more harm than good. And there's also peripheral inflammation, which is activation of the immune cells in the blood. And there's lots of fantastic work we've been hearing about, looking at this in animal models, looking at this in cells, looking at this in post-mortem tissue. And what I try and do is capture some of that really useful information and see how we can translate that into people living with dementia now. And some of the ways we do that are with brain scans, so MRI scans using magnets, or with PET imaging. So that's where you inject a radioactive dye with a specific tracer that finds its way to the brain, for instance, to try and capture these microglia in the act by recording sort of radioactivation that's emitted from these specific dyes, or by taking samples of people's spinal fluid and looking for inflammatory markers in the spinal fluid. It's a little bit easier to capture the peripheral information, because for that we just have to capture a blood sample. And that's why the work of the Biomarker factory we just heard about, who we work with, are so fantastic. So they're doing fantastic work at trying to get some of these brain proteins captured in the blood. So what does my sort of typical week look like? So on a Monday, I tend to do an NHS memory clinic. So that's people who've been referred by their GP or family doctor with memory problems or other kind of difficulties, come to see us in clinic, and we spend time with them, interviewing them, hearing about their symptoms, doing neuropsychology and cognitive testing, and organising scans or other tests to see whether we can make a diagnosis or refute a diagnosis of dementia. On Tuesdays, we typically have lectures and teaching, so spending time learning more, learning from different cases, learning from guest speakers coming in. On a Wednesday, this is often when we have research visits, so there should be participants coming not for NHS care or treatment, but coming in who volunteer to give their time and give their samples to research studies. And they'll come in to do scans, do blood tests, or lumbar punctures so we can get spinal fluid. Thursdays, I'm often visiting these participants at home, and this is a really, truly joyful part of the job, because I sort of drive around the county, finding people, particularly those with rarer dementias like Lewy body dementia. You might have to travel quite a distance, and meeting them in their own homes, telling them about the research we do, and seeing if they'll take part and be able to help out. Fridays, in an ideal world, I'll hopefully get to actually analyse, try and make sense of some of this data that we've been collecting over the week, but often it does end up being more admin. So how did I get here? So I came at this at a slightly different angle to lots of people, and I work with a team that is made up of people who come from psychology, nurses, technicians, engineers, a whole range of different people who work together on these big studies and are all essential parts of the team. the role I play is as a doctor. So I started by going from A levels to going to medical school. And some medical schools have these intercalated degrees where you can spend a bit more time doing research or a project. And at that time, I desperately wanted to be a cancer researcher. So I was very focused on doing cancer research. Following that, you can apply to do these jobs in the NHS where you do your general doctor training alongside training to be an academic and a researcher. And these are very highly sought after jobs and very prestigious, and I didn't get one. So I carried on doing my normal doctor training, this bit called the foundation program. But there's another stage where you can come back in and do that sort of joint clinical and academic training path. And that's another job that I didn't get. So I carried on my medical training. I did another 2 years of general medical training. And then finally I got a job, which is what I'm doing now, where I'm specializing in psychiatry, but also working for the University of Cambridge on these dementia research studies. So that's my job now as a clinical research associate. I don't want people to worry too much about the sort of pathways, because I'm sure it will all change, or it might be different where you are. But it just gives a sense of the journey often has a lot more barriers and a lot more twists and turns that you expect. And the destination might not be where you plan from the start, but you end up in a good place. So I think that's me. All right, thank you very much.
Thanks so much, Peter. That was a fantastic talk. And I think it's a really good message that we've all kind of faced rejection at some point during our career, and you just have to keep persevering, and there's always a way to kind of get where you want to be. So as I mentioned before, if you have any questions for any of the speakers so far, please put them in the Q&A box, and you can also vote for questions which you would like to be asked, so that they kind of flash up more for me. And yeah, next we're going to move on to our last main speaker of the event, which is Professor Mina Ryten. So Mina is the Centre Director and a Principal Investigator of the University of Cambridge UK Dementia Research Institute, and her group studies neurogenetics. So we're going to hear more about the science and also careers from Mina.
Thanks very much for asking me. I'll share screen and hope that that works. I don't know that it's... Okay, good.
Yeah, I can see that now.
Okay, brilliant. Okay, so I'm going to talk to you about making maps to navigate brain diseases. So right now, my job is actually based in London at the UCL in Child Health. So you might think, what is going on here on a dementia talk? And also as a consultant in clinical genetics at Great Ormond Street. So I'm going to start with this slide, and you might be wondering why I would put up a slide. Oh my God. Why is it doing that? Why I would put up a slide about a live worm being found in an Australian woman's brain as a world first. You can see I love the picture of the neurosurgeon who found the living worm. And then, there's obviously the picture of the brain and this very nice squirmy worm. And I guess the reason why I'm telling you about this is because that worm took a journey. And while I'm not necessarily sure that I had thought this analogy through enough, but I would say that to some extent, my journey has also been quite complicated. So the thought is that this worm was originally destined to infect pythons, and it's supposed to not actually infect human beings at all. And it's thought to have got to the brain because actually it didn't know where it was. It was in the wrong host. It decided to wander into the lungs, wander into the liver, and eventually ended up in the brain. And in a way, my journey to brain disorders and neurogenetics is not too dissimilar. So actually the very first piece of research I ever did was actually on placental tissue. So that's afterbirth for most of us. That's the bit that you don't want from the baby, the bit that's born after the baby's born. And then actually I decided, I might actually quite enjoy a bit of cardiology. So I was supposed to do a PhD, which was focused on blood vessels and the flow of blood through and the inflammation that can create in the wall of the blood vessels and heart attacks. Then that wasn't really working out. So I ended up studying skeletal muscle and the role of ATP signaling in skeletal muscle. And then somehow or another, I ended up in the brain. So a bit like this worm, I'm not sure I knew exactly where I wanted to go. And I ended up sort of somewhere interesting nonetheless. So in terms of that journey, I actually started, I'm really sorry about the jumpiness of the slides, but I'll try and keep it together. So that journey started actually, this is my little kind of worm diagram. So I started in 1995 as a medical student in Cambridge. And then I did an MD PhD course, so exactly what Peter just described, in fact. And then that was where I did my PhD on pure energetic signaling. So that's signaling of a molecule called ATP and its role in the development of skeletal muscle. And then I went back to finish off medical school, became a junior doctor, did kind of one of those academic clinical fellowships, and kind of still wasn't actually working on brain all the way through this time. And it was actually only when I ended up doing a postdoctoral fellowship on biomedical informatics with John Hardy at University College London, and that was around 2009, 2010, that I actually started working on brain at all. Even then, I was a bit distracted. I was like, okay, well, brain's really interesting, but I don't think I want to be a neurologist. I don't want to be a brain doctor. I ended up being a clinical geneticist instead, and then went to work for Genomics England, and then qualified as a consultant in clinical genetics, eventually becoming a researcher at the UCL Institute of Neurology, and then a professor at the Institute of Child Health. And now into 2024, it's quite a lot later, I've ended up as director of the Cambridge DRI. Now, you might wonder what's special about these orange circles as opposed to these blue circles. So a bit like Tara, it is possible to do other things other than do science. And so those 2010 was when my daughter was born, 2012 was when my son was born, 2021, and actually that should read 2023 is the second, these ones over here. That was when we got our dog, and that's when we got our second dog. So I thought I'd put the dogs on as well. So what am I actually really going to talk to you about in terms of neurogenetics? Well, I have actually what I would consider a pretty unique job. In a way, I do the mirror image of my job in the two places I do it in. So in the research lab, we're often asking ourselves, you have a genetic change in a gene. How do I think it causes a disease? Okay. If I go the other way in a clinic, I'm actually meeting a patient and I'm examining them or doing tests on them or whatever else. And what I'm actually asking myself is, this patient, what genetic change do I think has caused this patient to come in and have to see me? Why have they got this disease? Okay. So in actual fact, I kind of do the same job, but from two different places every day of my working life. And I would say to you that an accurate map, and here I guess I'm talking about an accurate genetic map, has never really been more important. It will tell me whether I got my diagnosis right. Did I guess correctly for that patient? Do I have any idea how severe their condition will be? So is this going to be really horrible dementia that's going to progress really rapidly? Or is this going to be a slow course for this patient? And maybe if I'm really lucky, I might be able to say how to treat the patient. So I think this is going to jump again, but I'm still going to go with it. So I would say to you that map reading in general, so that reading of a person's genetic map to understand what conditions they have, is getting harder. And that's because we're testing more people, and there are more positions in the DNA which matter for your likelihood of disease. So, just to remind you, you've got the cell, you've got its nucleus. Within that nucleus is the instruction manual for making the human body, and that's your DNA. Every single cell contains that instruction manual. It's split into chromosomes that you can kind of think of chapters of a book. And within those chapters, there's a recipe for making everything you need to make a person. And those are the genes. And actually what we do now is we test a lot of people's DNA, and we don't just look at small parts of their DNA, we look at the whole lot. So now on the National Health Service in the UK, we will commonly offer patients a whole genome sequence. That means the entirety of their genome sequence. That is making more diagnoses, but it's also identifying for us more disease-causing genes and places where really our understanding of disease wasn't very good at all. So to cure a patient, is it enough to know a gene? Well, I'm going to give you this tale, which is about a condition called Brown-Violletto-Van Leer syndrome. This is quite an old condition, and it used to be called Brown-Violletto-Van Leer syndrome, and more recently it's been called riboflavin transporter deficiency neuronopathy. In a way, you kind of like the first name more. So it was actually identified first in 1894, and there are 3 different people who identified it, as it were. And they all wanted their name on the name of the condition, so you end up with this Brown-Violletto-Van Leer, this very long name. And then nothing much really happens, right? People know this condition exists. It's pretty unpleasant. It results in your motor neurons effectively dying. That means that you can't control how you breathe, move, hear, or swallow. And eventually the whole thing kills you. So it's a pretty horrible disorder, actually. And no progress in this had really been made until, in fact, we get to around 2010. And that progress is because we finally identified the genetic changes in this gene that cause the condition. We finally identify the cause of the disorder. Once we identify the cause of the disorder, because people already knew something about those genes, they knew that they are transporters of riboflavin, we actually had an idea of how to treat it. So people started giving these patients high doses of oral riboflavin. And you might say, is that a safe thing to do? Do you need a trial? Does it need to go on and on and on to figure out whether this is safe? Well, we know it's safe because in actual fact, every person who picks up their Kellogg cereal packet, and I put brown flakes here because apparently they have amongst the highest quantity of riboflavin addition to them, is supplemented with riboflavin. So this is actually incredibly safe, and it does improve the outcome for the patients. So it's an amazing situation. From 2010, in the space of a couple of years, you've really managed to actually do something for this group of patients. But to cure a patient, is it really always enough to know the gene? And here I go back to, in effect, what Tara said, which is that a brain is really complicated. There are billions of neurons, trillions of synapses. And it's actually pretty hard to go from an instruction manual with individual genes to say, you know, this is how a person is becoming demented. This is the way that occurred. This is the molecular processes that require that. It's a bit like asking yourself with your tantruming toddler, you know, how do I understand how this tantrum occurred? Like what, you know, I thought that, you know, I'd given you yogurt, you clearly don't like strawberry yogurt and you're throwing a tantrum on me. Do I understand what the strawberry yogurt did to you? But so you can see that it's actually pretty hard to reduce everything and to go from simple fundamental rules to reconstruct the universe. So actually my research, you know, I think really from around 2010 onwards has focused on what I would call an intermediate phenotype. So I said to you, I'm going between the DNA and the patient, but there's something in between, right? There's the brain tissue itself and the RNA that is contained in that brain tissue. And, you know, here it's really important to remember your biology and remember that fundamental rules of the direction of information transfer in cells. And DNA is that instruction manual, but DNA stays inside the nucleus. It doesn't go out of the nucleus. It doesn't really do anything as such. Okay, I mean, that's a simplification, but still. What does do things is when that DNA is transcribed and you get RNA. And the RNA, in a way, is the action molecule that comes out of the nucleus. And that actually encodes the proteins, the building blocks for making a person. So in actual fact, you know, you could say, well, look, if I'm trying to find the connection between genetic changes and a disease, maybe I should look to the RNA. So to interpret genetic changes, you actually need what I would call a pretty accurate map again, an accurate map of the RNA, what we call the transcriptome. And that's because if you take a strand of DNA, it doesn't exactly have a kind of, it's not like logging into Google Maps and it's told you, oh yes, you know, you can go and find a cafe over here and there's a hotel over there. There are no markers, right? We've had to learn where the genes are. And genes themselves, you know, are strands of, are encoded in the sequence of the DNA, and they have exons and introns. Those introns are chopped out, you can see from these little scissors. And that's, you know, when you transcribe the gene, you have this pre-mRNA, those introns are chopped out, and you end up with your messenger RNA, which is kind of your working molecule, as it were. So the question is where do you chop? Do we know where all those little scissors are going to be? Do we know where the gene begins? Do we know where the gene ends? We need to know that pretty accurately if we're going to understand genetic changes, because it makes quite a lot of difference whether that genetic change is in an exon, an intron, outside the gene, inside the gene, all the rest. And what I would say is that transcriptome builds, as I've learned over time, are a bit like working models. They're like any other kind of map. And, you know, probably all of us have used Google Maps and it sent us to the wrong place. And maps have changed in accuracy. So this is actually a picture here of the map of the world in the 5th or 6th century. It's rather European focused, you can see, and like Australia doesn't even appear. You can't see any America. You know, neither north or south, and it's pretty deficient, really. It was probably the best you could do at the time, but it's not a great map, let's be honest, particularly if you want to go to some places. And I would say that over time, our map of the RNA that is in human brain and human brain cells has been improving and improving and improving. And that's as we've got to understanding, as you know, Tara described to you, individual cells and the RNA that they make. Okay, that's the thing that makes a difference between one cell type and another. And we're even going down to the location of the RNA within a cell. And then we've also got ways of thinking about the molecular resolution. That's another form of map, which might be what regulates that RNA. How is it spliced? Where are those chopping positions? And what affects its regulation? So we've been working for quite a long time on trying to improve the map, because we think improving, in my group, we think improving that map is going to improve our ability to understand the genes that cause dementia and cause neurodegenerative disorders that essentially kill brain cells. So actually, one thing we've done is try to look at regions of the DNA where apparently there's not supposed to be a gene, there's not supposed to be an exon, nothing's supposed to be happening here, but yet RNA copies are being made from that area. And we did this experiment, and we found that actually, and this is what I'm showing you here in this plot, is if we order all the brain tissues, and the ones in yellow are all brain tissues rather than body tissues from other parts of the body like liver or spleen, things that we don't really care about in this meeting anyway. And what we can see is that actually these red areas of the genome, these red lengths of genomic sequence, where there isn't supposed to be any active RNA being made, those are the parts which have higher quantities of that in brain tissue relative to other body tissues. And you can see that here as well. So the map's not accurate in brain. And part of the reason why that map's not accurate is because we haven't had very good tools to study that RNA. We've been dependent on what we call short read sequencing. So this is where we take an RNA strand, so copies from the gene, and we try and find its sequence, but we don't find its sequence from beginning to end. We actually only sequence little bits of it. And you can see that sometimes those little bits are going to inform us about the difference between transcript X and transcript Y, all made from that first gene. And sometimes those little sequence reads are going to not help us at all, and we're not sure. And so that confusion can make for us not really having a very accurate map of the RNA being made in the brain. So here I'm going to take the example of a gene which has mattered a lot to neurodegenerative research, and that is GBA1. So here I put Ellen Sidransky's picture here. Sorry, I forgot to put her name. So Professor Sidransky actually has literally just of today been announced as one of the winners of the Breakthrough Prize, and that is for her contribution in terms of the understanding of the role of GBA, this gene GBA1, in causing neurodegenerative diseases. So it's a huge achievement, absolutely huge achievement. And you know what this gene does is this gene actually makes an enzyme, so it has a job. And that job is actually, it's a processing job. And if you have mutations in that gene, if you have mutations in both copies of that gene, you get a condition which is called Gaucher disease, which occurs and presents usually in children. So this is why somebody who apparently is at the Institute of Child Health can also end up in a job in the Dementia Research Institute. But if you have only one abnormal copy of this gene, it actually gives rise to a very much increased risk of Parkinson's disease, and particularly Parkinson's disease with dementia. So what did we do? We wondered whether this gene GBA1, it has an extra copy of it, this thing called GBA pseudogene 1. So you may not have heard of pseudogenes. Pseudogenes are kind of genes that look like genes but don't work. That's the idea of them, really, that, you know, in actual fact, you know, you've got this extra copy, and that extra copy has a mistake in it, and you've just got these mistakes building up in the DNA, and this is called a pseudogene. And the problem is this gene and its pseudogene have a very similar sequence to each other. So when you're sequencing short segments of RNA, you're not sure whether you should, you know, whether we're talking about GBA1 or GBAP1. So it's a bit like confusing London, UK with London, Ontario in Canada. You're not sure whether you were flying to one place or flying to another. So here we were asking ourselves, could GBA pseudogene 1 be complicating our understanding of GBA1? And to address that, we decided we would use long-read RNA sequencing, a new technology where you can transcribe really long strands of RNA and cDNA. And so what we did was take bits of brain tissue, so exactly those kinds of little lumps that Tara showed you so beautifully. We also took neurons and astrocytes and microglia that you may have heard about yesterday that have been grown up in a dish. And we extracted RNA from them. and we sequence the GBA1 and GBA pseudogene 1 gene to kind of see whether actually our map is accurate. And what we found was that no, the map for GBA1 and the RNA that it makes is not accurate. In actual fact, we identified a number of transcripts, so actually over 15 transcripts that are originating from the GBA1 gene, and that would be expected to encode a protein, this green bar, so coding novel, and that in actual fact, these novel transcripts do not actually, they're not actually known to us, but they are being made. And the next slide, what I'm showing you here is actually being made in quite large quantities in the brain. So what we can see here in the caudate nucleus is that if we separate transcripts by the different types, and you know, for this talk anyway, the most important thing for you is the green blocks and those light blue blocks, we can see that there's an awful lot of RNA being made from that particular position in the genome, which actually we didn't really know existed or could be of that structure. We wanted to find out whether this GBA1 actually made protein, and we were able to show that it could do, and that actually what it might do. So to do that, we predicted from its sequence the structure of the protein it would be expected to make. And this was done using a new program called AlphaFold 2 that actually is using sort of machine learning and approaches to try and predict structure from sequence. And what we found really was from this is that actually indeed the transcripts that were being made would not be expected to do the normal job of GBA1 of doing this glucocerebrosidase activity. So looking ahead, I'm going to have two slides on looking ahead, and then I will absolutely stop. Is looking ahead, I think long-read RNA sequencing is going to be required to understand other disease-causing genes in the brain. These genes are complicated. They are, to some extent, their structure and their regulation is what makes us uniquely human. And so it's not surprising that when they go wrong, you get a uniquely human problem of dementia. So I think that journey between DNA and patient still has a way to go. And I think long-read RNA sequencing is going to help that. And I think it's going to help that, and I'm taking the example here from another disorder, beyond a case of just diagnosing patients or understanding things in a lab. So just as Tara pointed out, and again, this is not my work, you know, goodness me, I wish it were, but, you know, that actually by understanding the structure of our RNA, we can actually use therapies to target the RNA itself. And so there is this condition, spinal muscular atrophy, which is, again, can present at variable ages, but is primarily a disease of childhood, a genetic disorder of childhood. And the gene causing that condition, SMN, was originally discovered in 1995. But by 2006, an RNA targeting therapy had been developed. And this has been very successful. Over 19,000 people have been treated. And so in a way, I guess what I want to leave you is the thought that it's never been more important to have a good map. People need to build good maps. So finally, I just want to thank the people who donated their brains to research. That is absolutely key to the work I do, and that's most important. I also wanted to thank the people that are in my lab. I love working with them. They're a great bunch. And I thought I would also put up our furry members. So we have our actual human members. Our furry members are people's pets from the lab. And this is my dog, Rocky, as well. So I just thought I'd put a picture of him because it's my lab, so I can show my dog. Thanks very much.
Thank you so much. That's a very cute dog. I'm kind of jealous. Thank you so much. Brilliant talk. And I think it's really nice to see that progression from, as you mentioned, like getting these clearer maps, being able to see so much clearly where these diseases are coming from. And that seems to be the case across a lot of neurological conditions. So really interesting. And if you have any questions for Mina, please do put them in the Q&A box, and we'll come to them at the end of today's session in about 15 minutes. So our next, what we'll have next is a Follow Me Around the Lab video. So this is our final Follow Me Around the Lab video of Making It Brain 2023. This was created by Gia Du at the UK DRI Imperial College London Centre, and Gia works mainly on stem cell models of disease. Dia, for making that video. I think that's a really nice insight in what it's like to be a wet lab scientist, particularly working with cells. And you can see that it's kind of quite an active job. You're on your feet and doing a lot of practical tasks during the day. So if that sounds like something you'd be interested in, you might make a great scientist. So for our final session of today, we're going to have two last flash talks. And these will be by two students from Blanca Diaz-Castro's lab in the University of Edinburgh, the UK DRI Centre. And Noelia is a lab-based research assistant. And Hannah is a secondary school student who actually did work experience in their lab this summer for a week. And this was arranged after Making It Brain 2022. So Hannah reached out to one of the speakers and managed to secure herself some work experience. And so we thought that this would be a really nice way to for you guys to hear about how she did that. So I'm going to let you guys start. I think, Noelia, you're going to go first and share your screen when you're ready.
So I'm going to tell you a bit about how I got to the lab and a bit of what I do, okay? So first of all, I started by wondering what are my interests. Well, when I was a child, I really liked riding bikes, but that very same day, I fell off the bike, so not great at it. I also really like plants, but all of them die within a week. So then I thought, why not being a scientist? I promise I'm much better at it, I promise. So I went to Scotland. I'm from Spain. I went to Scotland to study neuroscience with psychology. And from the very beginning, I chose a placement in my studies. Then COVID hit, quite literally, as you can see. And I thought, well, that's even better. Now I get to have, practical experience, which wasn't quite easy with COVID. So looking for my interest again, I found Dr. Diaz-Castro lab. I contacted her and she found me very suitable for a research assistant role. And well, you might wonder, what's my role? I think it's learning, just like that in general. So we have a mantra in the lab, which is look, do under supervision, and then do it alone, right? So I started by helping everyone out and learning the techniques that way, learning analysis, but above everything, I would say learning to think like a scientist, which is very important. And then I move on to my own project. Well, that's me in a conference. So what do we do in the lab? So we study astrocytes in aging and AD. So what are astrocytes? Astrocytes are cells that tile the CNS, and they contact multiple cells like neurons, microglia, oligodendrocytes, and the vasculature. And they have shown to suffer changes through AD and aging, and that's quite literally my project, studying changes in astrocytes throughout age and pathology in a mouse model that has some of the characteristics of AD. And this, can you see? This green thing is an astrocyte, and that's a blood vessel. Okay, so very easy take-home messages. Find your passion, okay? Learn as much as you can. Be kind to yourself. You're gonna make mistakes. It's fine. You're just learning. Especially don't give up and, follow that mantra because it really works. Helps you out to not be so lost in the lab and ask all the time if you have any doubts. I wanna thank everyone, especially this great team, to which, as they've mentioned before, Hannah joined. And well, now she'll tell you a bit about her experience in the lab and you'll see. Thank you very much.
Okay, so thank you for the introduction. My name's Hannah. As mentioned, I was in everyone's position who's watching here this time last year, and I was really taken aback by how amazing this conference was. So I'm a year 13 student now, but I was just starting year 12 then, taking biology, chemistry and Spanish as my A-levels. And at the point last year, I was a bit unsure whether I wanted to do medicine as a career, or if there was another pathway that I wanted to go on. So amongst the amazing speakers was Dr. Blanca Diaz Castro, and her research and her career sounded incredible to me, and I began to think about how perfect a neuroscience career would be. So when it came to finding work experience, I thought it'd be really good to contact her and just say how much her talk on this conference helped me in deciding what to do. And she was so welcoming from the start, and obviously offered for me to come and do my work experience week. So this is just a run through of what we did. So on the first day, we observed Holly doing a PCR, which is basically testing for genotypes of the mice that they use. And then Blanca talked through with us the two experiments that we were going to be doing, which is the immunohistochemistry, which stains and dyes the blood vessels and astrocyte end feet, and also the protein pull down, which basically isolates the proteins from the astrocytes and gets rid of all other brain cell proteins. So day 2, we started off by reading a bit of a research paper, and then we finished the immunohistochemistry experiment. Day 3, we made buffer solutions for the protein pull down, and we began to do the protein pull down experiment. We also started looking at some of the immuno results from our first experiment, which is the furthest photo, the 2 photos at either end. So here you can see they're all dyed separately. So the green bits are the blood vessels, and then the orange is the astrocytes and their end feet. So it's really cool. You get to see it in 3D and see all the different parts. And then on day 4, we carried on with the protein pull down, and then we also got to go and see a piece of human brain tissue, which I think was one of the coolest things I've ever witnessed. And finally on day 5, we started with Journal Club, which was really cool. We got to talk through the research paper that we were reading the rest of the week and analyze it, and it was really interesting to see scientists talking about other scientists' work and discussing the issues around it and what was good about it. And we also finally got our final pictures of the astrocytes and their end feet. So the picture with the pink dots, that's the nuclei, and then the blue bit is the blood vessels. We've got a massive blood vessel where the black dot is, and then more astrocytes. And also the other picture here is a slice of mice brain on a slide, which we were using to look through, and I just had to include that because if you're able to zoom in on the photo, you can see the actual slice of brain, and it's incredible. So just to summarize, the work experience week was amazing. Definitely an experience that I'll remember for the rest of my life, and it really made me realize that this is the career I want to do. So my next steps are to keep working hard with my A-levels. So I'm just finishing my uni application now, and Blanca was absolutely incredible, and she's certainly a contact I'll have for support for the rest of my career. Also, thank you to Holly, who was helping us in the week that we were there. They were all incredible, and I really learned a lot and got to experience things that I never thought I would get to experience. So thank you for listening, and I hope that if you're inspired by this, don't be scared to contact any of the speakers, because they're all lovely people and you will get some amazing experiences.
Thank you so much, Hannah. It's so great to hear what a positive experience you had, and also how inspired you were to kind of pursue neuroscience in the future. So I hope we can do that for some of the participants in today's webinar as well. So if you have any questions for Hannah or Noelia, please do put them in the Q&A box, as we're going to shortly start the Q&A session. Just before, I want to remind everyone that today and yesterday's Making It Brain sessions are being recorded, and the recordings will be available on YouTube. And everyone that registered will receive the link to the recordings in their inbox. And just as a last bit of admin, I'm just sharing my screen. What you will also receive in your inbox is a list of links and resources about careers in neuroscience and also about neuroscience research. So this list has been curated to include lots of accessible things, including diary entries from people who have careers in neuroscience, but also more information about learning about Alzheimer's disease and other neurodegenerative diseases. So everyone here in the call will receive that link. And finally, we will be sending out a feedback form about today and yesterday, and it will only take you 1 minute to fill out, but it's so valuable for us to have that data so that we can curate an even better Making It Brain next year. So please do fill that out, and everyone again who registered will be emailed that link. And before I begin the Q&A and before I forget, thank you so much to everyone that helped Making It Brain possible this year, including the headquarters team of the UK DRI, all the people behind the scenes who helped you all find Making It Brain, and especially to those who made the Follow Me Around the Lab videos, which I hope you all really enjoyed. And if there was any issue with any of them, they'll all be part of those recordings with hopefully some higher quality videos as well. So now I'm going to invite the speakers from today to turn on their cameras if they can and join us for the Q&A session. And if you have any more questions in the audience, please do put them in the Q&A box. We have a lot already. So it's great to see such high levels of engagement. So one of the first questions that I wanted to ask that I saw in the box here was, well, quite a few questions have actually asked about undergraduate degrees. So people thinking about pursuing undergrad and not quite sure what they want to do that in. And some people have mentioned, is it possible to do a degree in psychology and then pursue a career in neuroscience or cognitive neuroscience, medicine, and also computer science? So what I wondered if the panel could do is maybe comment on the undergrad that they did, and also if anyone is hiring staff, kind of mentioning the types of degrees some of your staff have done, if it hasn't been kind of a pure neuroscience degree. And so I'm thinking maybe Tara, we could start with you as you're at the top of the screen.
Sure, thanks. Are you hearing me okay?
Yeah.
Great. So my undergraduate degree was in biochemistry and French, keeping in mind that in Texas, 20-something years ago, there was not a neuroscience degree. I've hired people with degrees in undergraduate neuroscience, undergraduate biology, and undergraduate anatomy, and they've gone on to do PhDs or masters in neuroscience. Neuroscience is a broad church. We have people with lots of different skill sets, from computational to wet lab. So do think about any career any degree that really fascinates you. If it's related, you can probably transition into neuroscience after.
Great. Thank you, Tara. And Peter?
Yeah, I think following on from that, it's just it is really broad. So I did medicine as my first undergraduate degree. I think the difference with that is it's a professional qualification that makes you into a sort of clinical practicing doctor. And so you do form a sort of a specific part of the team, which is as a clinician. In the clinical side, we work a lot with people who have undergraduates in psychology as well as neuroscience, and then go on to specialize into dementia research. So yeah, it's really broad, and it depends on sort of partly what area you want to be working on and where your interests are.
Brilliant. And Mina, could you also comment on that?
Sure. So I think, so yeah, again, my undergraduate was in medicine, but, you know, I don't know that it was the most exciting undergraduate, to be honest. But I mean, I think, yeah, you're nodding, Peter. So I think in terms of people who are in the lab, lots of computer scientists, actually, you know, sort of a lot of those come to my lab, people with biochemistry degrees, people with some with neuroscience degrees, but I think maybe only one, actually, to be honest, thinking about it ever. Also on my side, maybe genetics, and you know, other routes people have gone down is things like clinical genetics masters or genetics counselling, but maybe that's because I'm a more genetically orientated sort of crew, really. But yeah, so I think you do what actually suits you, and it's hard to give a one answer fits everyone. And probably that French part of your degree, Tara, was probably quite good. I don't know whether it gets used now, but it was probably made you happy.
Yeah, I think that's really good advice as well. Do what you're actually enjoying, and there's always different ways and different avenues to get to the next step. Noelia, do you have any advice on that one?
Yeah, so I studied neuroscience. because, well, in Spain, we really don't have that opportunity. So I moved to Scotland actually because of that reason. I was going to study medicine, but I find it's a quicker way, if you might put it that way. I'm more like lab related, as Peter mentioned before, to get into neuroscience and that kind of stuff. But actually, you can study other degrees like I don't know, immunology and that kind of stuff, and still work as a neuroscientist, because there are so many aspects that you can work on in lab that, well, it just depends where you apply to.
Noelia, can I just say one thing about that? Because I think that's a really good point about things like immunology. I mean, as Tara was saying, and I think Peter said as well, actually, it's not as if, and maybe I'm the clinical geneticist, not like these things this way, but I mean, it's not as if other bits of the body don't contribute to your brain health, right? So is it particularly obvious that, you know, you should be studying this if that's the future of the disorder? I mean, you might argue that right now it would have been a good bet to study immunology to be the person to study dementia, right? That's what I feel I should know more about.
Yeah, I totally agree. And I think as well as those kind of fundamental principles of the subject you're doing, a lot of those degrees will give you really good experience in a lab, and so many different jobs and so many different research groups come down to the basics of lab work. So I think you can kind of go anyway. And just for myself, I started a degree which was called General Science, and I was able to kind of tailor modules towards myself and eventually specialize in neuroscience. So lots of different degree programs will have lots of different approaches, but you can kind of tailor things to your own wants and what you find interesting as well.
Actually, sorry, if you allow me to give just like one small advice, is that whatever you do, just don't forget that we do this for people. So we don't just go to lab, or we do it because of patients, because of people that suffer a disease, or because we want to understand more about the human body. So that's something to keep in focus, I think, for all of you.
Yeah, and there's so many different diseases out there that need attention as well. So kind of as we've touched on more of the science side of things, there's a question here that says, what would you say is the reason we are seeing such an increase in neurodegenerative diseases? And I was thinking maybe Tara, you could start with that.
Sure. One of them is our aging population. So luckily, research works, science works, and a lot of things that used to kill us off early don't so much anymore. So we're better at taking care of our hearts and treating cancers. So we're living longer. And remember, I had those 3 buckets: age, genes, and lifestyle. We're aging. So that's one of the big ones. But we don't know all of the causes. So it's a very good question. Come and help us find out.
Would anyone else like to kind of contribute to that question about why we see more of these diseases now?
I mean, I think Tara's point is, I mean, I think it has to be age as being one of the major components of this. I mean, also, we're probably not leading very healthy lifestyles, right? I mean, you know, that's I mean, I'm sure that kind of things like obesity and with it kind of, you know, the kind of risk of diabetes and cardiovascular disorders are going to be doing nothing for the quality of your brain. I mean, I think people, you know, need to kind of do the basics of eating well, sleeping well, having their friendships around them, and feeling kind of comfortable in the place they live.
Yeah, and there's kind of a phrase going around, what's good for your heart is good for your brain, which is good to keep in mind for kind of general well-being as well. And there's another more science-based question that I'm seeing coming up for Mina as well. So are all these neurological diseases based on a genetic mutation?
No, is the short answer. So I mean, the thing is so actually, if you get to things like Alzheimer's disease, actually very few patients will have Alzheimer's disease because of its single highly important genetic mutation. So actually most people will have a little bit like almost a lifestyle issue. Most people have little risk positions all over their DNA that have increased their risk a little bit, but are not actually a single place in the DNA which you could say that is the cause of you having this condition. However, you know, it does vary between disorders. So for motor neuron disease, that's not quite so true. A quite considerable number of patients who present who apparently have no family history will actually have an underlying genetic cause. So I think the statistics range between 15 and 20%. So, you know, those kinds of things are important for a Parkinson's patient. If they have had it, if they present with Parkinson's early, if there's a first degree relative, there is quite a high likelihood that there is a genetic change underlying it. So it does differ by diseases, but I think most important about those discoveries, and I think Tara touched on it in terms of therapies, is that those have actually given insight into how the disease is caused, even in the common form of the disease. And consequently, they have been key to driving research in the area. So these newer therapies are really about amyloid clearance, and that's very much based on the identification of mutations in the amyloid precursor protein, giving rise to early onset Alzheimer's disease. And yeah, I think that's, and just to say maybe for Peter as well on the psychiatric side, I really think that there's been a mentality in some areas of medicine where, you know, diseases just happen, you're not going to be able to do anything about them. And I think that was the attitude about old age diseases and many psychiatric diseases. I think what genetics does is make them feel like they're things that you can treat, they're things you can do something about.
Thank you so much. I have a question for Hannah. So Hannah, where are you planning to apply to university? And what advice would you give to other year 13 students or kind of final year school students writing their statements for university?
So currently my what's been my dream to apply for a while is University College of London. But I'm currently in the process of looking at universities. So I visited Bristol last weekend and we're visiting Cardiff this weekend. So we're all over the country at the minute. So I'm not sure about that. I'm trying to find somewhere where I can keep Spanish in there and maybe do a placement in Spain, because obviously I'm doing my A-level in Spanish, so that's still really important for me. And then in terms of advice for writing personal statements, I'm obviously in that same boat right now. I'm trying to get mine done, trying to find the way to write it the best. I think just put what you think is you and be really passionate about the subject that you want to study, and don't be afraid to sound like you're bigging yourself up, because that's what they want. And even though sometimes I think we all think, oh no, I can't put that sounds really like vain, but you need to put what makes you, like what sells you. So yeah,
I think that's great advice, making sure you recognize your good qualities and all the hard work you've done and really shining a light on that. So that's great advice. And then I'm going to ask the panel one final question. So quite a few questions in the Q&A are asking for advice about how you would recommend trying to gain research experience, both as a school student and as an undergraduate student, and other types of tips for strengthening your CV for maybe future applications. So maybe everyone could have a little bit of a think about kind of what advice they would offer people if they want to get work experience. Does anyone volunteer to start answering that first? Yeah, Noelia?
So, well, in my case, I did a summer placement and now a full year placement. That's really helpful. And sometimes they can be paid or unpaid. It depends on, your needs. Especially, I'm not, everyone knows their situation. So, but it's very nice to gain any kind of experience. And also just, try to look out for things, talk to people, talk to your lecturers if you're interested in like the topic that they teach you. If you, I don't know, if you read about someone, just try to contact them. They might not answer, but if they do, they might offer you, I don't know, like Hannah with Blanca, and you never know. So it's always good to try and just don't be shy. I think we all understand what it is like to start as, a university graduate, or just even before graduating for university. So it is just normal. It's science. So
yeah, and don't be afraid to contact people, because the worst thing they can do is say no.
Yeah, pretty much.
Great. Who'd like to go next?
Oh, I could say something. So, I mean, I remember I got my first experience was basically I wrote to a load of people actually at UCL, thinking about Hannah, and asked them for a job basically for over the summer. And actually, I got quite a lot of no's, or actually just no answers really. And it was the kind of time when you wrote people a letter. You didn't even actually, so you posted this thing and asked them. And actually, there was only one person who said, yeah, why not? And that was Jeffrey Bernstock, who said, yeah, you know, I need someone to go and collect placental tissue out of, you know, outside of, you know, the UCLH's maternity unit. Can you go and, you know, so basically I spent the summer waiting for afterbirth. So that's, I don't know, you have to start somewhere.
And it's all great experience and kind of helps you get to where you need to be next. Tara, do you have advice for how to find placement, but also maybe advice that you would give based on someone who takes people in on placement?
Yeah, I mean, I think you've covered it. I think that Noelia and Mina, don't be afraid to ask. Some of us might not have time to respond to all of the emails in our inboxes, so don't be offended if we don't, but we try our best and we're nice people and we do like to help people. A little bit of different advice would be if you can get into a program, like I work with Into Science, they're amazing. So if you can show the initiative to apply for a program like that, or I'm sure in your local area, if you look through things like the British Neuroscience Association in the UK or the Society for Neuroscience in the US or FENS in Europe, you can maybe find programs that will have internships or sort of facilitate mentoring, and that can be really powerful. So show some initiative, do what you think is really exciting, and do just get out, put yourself out there, and don't be offended because, like Peter pointed out, a lot of our job is rejection, and that's fine. You just have to sort of, it's not fun, but it's part of it, and it's fine to put yourself out there and keep trying and get rejected. It's all part of it.
Yeah, great advice. And I've just put the name of those programs into the chat. So Into Science is for school students looking to get involved in 1 week work experience programs, and Into Research is for university level students looking to do summer internships in research. So they have websites for those that you can find. Peter, would you have any advice, or do you have any experience in taking on any students?
No, I think it's very similar to what everyone else has said so far, in that I would write emails out that said, you know, this is what I'm doing at the moment. These are the things I can do. I'm interested in learning this, and I see that your group are doing this. Would there be any opportunity for me to do something like that with you? And for lots of supervisors or PIs or whoever, it's just not the right time, or they don't have any projects like that. But then you will find someone who, you know, it is the right time and who is interested. And so I think being clear about what time you can commit as well. So you can say, well, I've got a 4-month block here, or over a summer, I've got 2 months here. So someone can say, I've got a sort of a 2-month size project to do that would fit with you. So just sending, you know, real clear, this is what I'm interested in. This is what I can do, what skills I do have, and this is what time I have. Do you have anything that's suitable? And then, like everyone else has said, a lot of people you won't hear from, and it's just because, you know, inboxes are full or they're busy. It's not the right time. But then you'll find someone who sort of has that overlap in interests, and will have something for you to do.
Yeah, and I think.
I was just going to add to that. I think one thing actually that you know, where Tara's point about if you can get on a program, do, is because I think it's become, I mean, I think this is a good thing overall, but a lot of the universities have much more set rules about how you can hire someone, the process of doing that. So it can actually be more difficult than you imagine, even if we have an opportunity to say that, you know, you can create that for somebody. So, I mean, particularly if it's paid work, that has sort of hefty complications to creating a role. So, and I think, you know, in the time when I applied, I think it was just easier actually for a PI to do that kind of thing.
Yeah, and one more piece of advice that I would give that was shared by Joe Jackson yesterday is that if you are emailing someone and you want to be interested in their, make sure to express that you're interested in their work specifically. You don't want to write kind of a blanket email and send it to 100 different supervisors, because that type of thing gets passed over much more quickly. But if you have a genuine interest in someone, then it's more convincing if you apply to them directly. And just to mention that on the UK DRI website, all of the group leaders are listed on there, and what they do and what they work on. And so that can be kind of a first pass look at maybe if there is a centre close to you, you might be able to find someone through that. And Hannah, I noticed you just unmuted, so if you want to give us one last word.
Sorry, I just wanted to say as well, everyone's like covered the advice really well, but one more thing is just don't be disheartened by the no's. Like I've been there, I emailed so many people like thinking, oh yeah, this would be really good. This person looks really cool. This research is amazing. And there's either no reply or there's a no. That's the majority of the time because these people are busy. And but just don't be disheartened by that. Keep looking for someone, and there will be someone who wants to take you and wants to teach you. So just keep going. And yeah.
Thank you so much, Hannah. I think that's fantastic advice to end on. I want to thank all our panelists here for staying and answering the questions. Thank you to all of our attendees over the last 2 days. And a huge reminder that's really helpful for us is to fill out the feedback form that will be in your inbox shortly after the event. And it will help us shape Making It Brain next year. And the recordings of these recordings of these sessions will be available on YouTube after the event. So if there are any details you missed or anyone's contact details that you would like that were on their slides, that will all be available on the recordings. So thank you everyone for presenting, and thank you to all our attendees. I think everyone really appreciated your insight.